topology with many students doing PhDs in this field. So I meant to be in the first part is a very gentle introduction to the ideas in topological data analysis. And part two is mostly what I have been doing. And it's related to this somehow, but not fully. So, so this is two-part talk. So the first part is data comes as sampling as a bunch of points in Euclidean space, if you can make it so. This is called vectorization. So you can get lots of data. It could be graphs, it could be connections, matrices, and then you make them into a sampling of a metric space. And then what you would like to do is to get topological and geometric information from these spaces. Right. Let's see why it's not working. Okay. So that turn raw data into measurable or quantifiable metric data, then use topology to process it, because topology uses metric spaces as an input. So the shape of data, this is the title of a very famous paper by Gunnar Carlson, where who started this in the early 2003, 4, 5. So the idea is, so you have data which is clustered maybe around some shape, and you want to know what the shape is going to be. So in this case, you have data which are a bunch of points, and they make up some kind of a circle. Of course, you can see it because you don't have that many points to deal with. You don't have that many points to work with. But generally, you have thousands of points. And just taking points on their own, there's no topology. It's very, very interesting. It's completely trivial. But what you want to do is associate to these points some sort of a space that you can study and that can give you information. So I'm going to explain this idea, which has been very successful. It's called this persistence, in very simple terms. So I hope people will enjoy the illustrations. All right, so you have a bunch of points. So you have to think, of course, that generally it's thousands of points which represent data. Here I have only a few. And what we do is choose a distance d for diameter and cover the points by disks or balls, if you want, in higher dimension space of that radius d. So the center of the balls are the points themselves. All right, so you have as many balls as you do have points. So for a given d, you can cover up this. You can thicken, if you like, the points, the data points, into something that looks like this. And now you connect pair of points. So you start connecting the points. So you start constructing an underlying space by doing something like this. So you connect pair of points that are no further apart than d. So if the 2 points are farther than d, you don't put a segment between them. But if they are closer, have a distance that is less than d for that given d, you put a segment. So here you see, I'm not sure if you see the cursor, you see that this distance between these 2 points is less than d. So I did put a segment here, and I did the same thing with all other because it just worked out to be okay. All right. So now what happens? You have overlaps. But before I do that, yeah. So for d small, you're going to get this picture here. So you're going to get a certain number of disks then covering the of a certain diameter. You can see some topology here. There's something interesting here. But for
Sadok, peux-tu réactiver le son? Répète, répète.
Okay. Can you hear me now?
Yes, yes.
Okay, so can I go back, or is this okay?
It was just perhaps the last sentence.
All right. Okay. So let's say I am picking a diameter d, and I'm covering the ball, the points, the data points by these balls of diameter d, and of course, depending on the diameter d, I'm going to get a space which is a union of balls. The union of balls is not so interesting. I'm going to come to something slightly more interesting, which deformation retracts to. But whatever the case is, on the left-hand side you have a small d, so you can see here there's some nice topology. So at least you see a connected component here. You see another connected component here, and you see one single connected component. Here you see them all merging, and it's not so interesting. So the whole point is how to choose this distance d in an interesting way. In fact, it will, you will not have to choose it, as I'm going to explain. All right, so we're going to consider all the distances. So for every d, I'm going to do this construction with the one that I started explaining. So say I have 4 points like this, and I take a disk of radius d. And as I said, because the distance between the centers is less than d, I'm going to put a segment. So I'm going to have the segments. You see, the union of segments is like a circle. And the union of the disk also deformation retracts on the circle. So replacing the union of circles or the union of balls by something slightly more combinatorial is going to be very interesting. But then you have to be careful, because then you might have triple intersections. So if you have a triple intersection, what you put in is, so for example, if the three disks here, tu vois le curseur, Nabil? No. Oh, okay. Oh, okay. Oh, how can I do that? All right. Anyway, so if you see the three disks on the right hand side, and this is for D2, so D2 is slightly bigger than D1, so the disks are a little bigger, and so they overlap. They were overlapping on top, so they're overlapping on the bottom. But now we have triple intersection. So when you have triple intersection, you fill in. So now you have three edges, because all of the three, I'm looking at the three disks on the right-hand side, and the same for the three disks on the left. But now, because there is overlap, all three of them are overlapping, you fill in by a triangle. And now, again, if you have quadruple intersection, you put in a three simplex, which is called the simplex. So you move on like this. So, so at the end of the day, for each D, you obtain a simplicial complex. All right, so here's, I explain again. You see here how can I get the cursor here? 1 second, maybe pointing this pen, laser pointer. Now.
Yes, now, yes.
Okay, all right, well, that's fantastic. All right, that's good, that's good. All right, so see here, I do not have the distance between these two guys is bigger than D for that particular D, all right? So I don't put an edge. Here I put an edge because the distance is less than D. But here I have three overlaps, so I put a triangle, all right? Here I'm going to put a bigger tetrahedron, so you don't see it because I have higher overlaps. So the gray space you see, it's called simplicial complex because it's made of simplices. So it's a bunch of triangles in higher dimension. It's more combinatorial. It's very clearly, it's called the simplicial complex. Is the first thing you learn about if you do TDA. Something extremely combinatorial. It has vertices, it has edges, it has triangles, it has tetrahedron. It goes like this. And you can put it on computers. You can algorithmically deal with this space like this, and much better than the union of balls, but you can show that this union of balls is going to deformation retract onto the space. So, okay, so this for the left, I have a little small d, on the right, I have a bigger d. And then what's the idea is that for various d's, I'm going to get various I'm going to get various simplicial complexes. All right. So here I have one complex for a smaller d. I get bigger d. I get this one, and I get. Of course, I have inclusions because if I have overlap here for small d, I certainly have overlap for bigger d. And I have a sequence of inclusion like this. So I'm getting d to vary. So again, this is all starting from a cloud of points. So I'm starting with points. The points are the vertices, and from those I am constructing spaces. And so now I'm look at every feature. There are many features we can deal with in topology. It's called homology. I'm not going to introduce homology to you. It's insane to be able to do that in a talk like this, a 50-minute talk. But it's very simply this H0. It's a group, and the rank of that group is the number of connected components. So what I'm going to do is, for this space, for a given D, I will look at H0, the number of connected components here, and I see how the number of connected components is going to change. All right, so the number of connected components here is 3, because they have 3 connected components. Here it's 1, here it's 1, and it continues to be 1. Now I'm going to look at H1. So if you want to think about it in a very simple way, H1 is an abelian group, and the rank of it, say it's the number of circles that you can see that do not bound a simplex, right? So here, for example, here this is a circle, but that's not interesting because it's the boundary of a thick triangle, right? But here's this bigger circle. See, this is a circle, right? This, if you think about it, is a circle, right? So this is going to contribute B1. So it's going to contribute to B1. Here, there's no B1. Oh no, there is a B1 here. Here. There is a B1 here. Here's a circle, something that's homomorphic to a circle, and it's not a boundary. Here's another one. So what you do is you look for B1 here, B1 here, B1 here, and you continue looking for B1s all through the sequence as D goes from very small to very big. Okay, that's the idea, right? And it's a very potent idea, right? So here I'm explaining again. So here for very small d, for d equals 0, you have only the points you have started with, then you get some bigger diameter, so you get bigger balls, so you put them in. Here B0 3, because as I mentioned, exactly, you get 3 components. Here B0 is 1. But see, the B0 is 1 is the one that persists, meaning, and that's important for later, is that I can get there's some circles or some connected components appearing and disappearing fast. But what I am really interested in is those that will last for the larger possible values of D. So the persistent values, the persistent features are the ones describing the geometry underlying these cloud of points. So for example, here I have 3, so you see here I have 3 connected components. So this connected component is born because they're all connected up, but then when D is bigger, it'll die. It will merge with another connected component. So there's this notion of birth and death. I have to look at the time here because I have a second part I want to get to. All right, that's fine. So you do the same for H0, H1, H2. I hope it's clear. I'm going to have one more illustration just to explain this a little more. All right, so this is what we were saying. All right, so you see here for this particular D1 at this particular time, at this particular D1, all 4 of them are going to merge, all these disks are going to merge, and suddenly they're going to make up a circle. So in here, I do have the birth of a circle or a B1. And now it has vanished. That circle is gone when I got from D1 to D2. So, I can visualize this by saying that there is birth of a feature, there's birth of a B1, an H1, a circle, right? At distance D1, a diameter D1, and it goes until D2. When it goes D2, it disappears, because now the circles are the boundary of triangles, and so they do not account for anything of interest. So this is, at the end of the day, what you are going to get by doing this for H0, which means the connected components. You do this for H1. You look at what happens, which are for the underlying circles that are not boundary of triangles. And then you look at higher spheres, which are not boundary of balls. And then what you are going to get is a barcode. So this is a barcode, for example, for this bunch of parts. So here's the illustration here for this particular data set. Okay, so here's the data set. You do this construction. You start by d equals 0. Okay, so I'm going to start with d equals 0. All right, so let's look what happens. You are going to put in disks of varying diameters, and you are going to measure up the B0, B1s, and B2. So what do we have here? We have at this stage, we have a birth of here where the cursor is going to be. Let me look at the B1, because B0 is generally not interesting, because you start B0, the number of connected components you start with is the number of points. So you start with as many. So if you have thousands, it's too many. So we don't look at B0. Really, we look at, we start looking at B1 and higher. All right, so let me see if I'm, yeah, so let's concentrate here on this circle. So you see here, just right slightly before one, I have possibly this guy has been created. So this circle has been created. And then shortly afterwards, this circle has been created. Right. Because you see, this deformation retracts onto a circle. Same here. So now in this barcode, I have birth of the first circle here, and I have birth of the second circle here. And now they are going to die at some point, because the diameters are getting bigger. This one is going to die. Maybe this one is going to die first, and this is going to die second. But what's going to happen? All right, let's see. All right, death, birth. All right, now I have the death of these two circles. This one is dead now, because it's not anymore, it's bounding something, so it's not interesting. The same thing is here. But now look, I have a much bigger circle that has been created here. So when all of these guys merged, and for this particular D, so for diameter between 1 and 2, 1.6 roughly, or 1.5, they all merge and make a circle, an underlying circle. So the data that we were looking at is really trying to tell you that geometrically behind it, there is a circle kind of a configuration. But please pay attention to this. This is the last thing that I want to bring up in this part. So the diameters are increasing. The circle is still there, right? Oh, when this vanishes, it's gone, right? And everything becomes a mush. At the end, you don't get anything. You get a contractible space at the end, because it's a union it's a ball. So basically, you're not getting anything. But the one that persisted, the element that persisted is this. These two guys are like noise, right? We are considered to be noise. The small barcodes are noisy data, and the ones that persist are what people study. There's a persistent features. Okay, that's very good. Is that clear? Okay. Nabila, am I going fast? Am I?
Okay, for me, there is many new things, but okay, go on.
No, it's new. I know it's new, and this is why I don't have any equation. I don't have any algebra. I only have pictures.
Okay, just a question, perhaps. What is the information here is the variation of with D, and not the values for specific D.
No, that's precisely the point. We don't know which D to pick. Right, we don't know, so we have to take them all, right?
Okay.
We take all D and all positive Ds, and we let D vary from 0, diameter 0. All right, so Nabil, when diameter is 0, the discs have diameter 0, so the discs are the points.
Okay.
So it's not interesting. So now you start increasing the D, and you start seeing what features, what topology, what is the topology that you're going to see by doing this business of constructing the simplicial complex, means relating here. I'm going to do it, right? So you see, when they merge, you put a segment, they merge, right? When they touch, right? So when they touch, you put the segments. If 3 of them touch, you put a triangle. If 4 of them touch, you put a higher, all right? So at this particular D, at 1.2 or 1.3, you have, what do you have? You have 3 connected components. Do you agree, Nabil?
Yes, of course. Yeah.
All right. So you have 3 connected components. By how many circles modulo boundary, which we call H1? How many circles modulo boundary? You have 2, only 2. You have this one because there's a space here. So because there is a space here, it's not being covered by the balls, you can deformation retract it onto the circle. Here, same, because you have space here, which, you know, you can deformation retract this to the circle. So you have one circle that has appeared in this process for a D equal 0.9 possibly. And at later D, you have another circle that showed up, but they are going to disappear because when these Ds get bigger, they overlap, and when they overlap, the circle disappears.
Okay.
All right, so say they disappear here, right?
Yes.
Okay, now this is no longer a circle, there's no B1, and here there's no B1. B1 is the rank of this H1, right? But this is the important thing, the 2 important things. There's another circle, this is called another B1, which, see, this is like a circle, right? If you make a deformation, if you deform the whole space onto this circle that I'm outlining, what I'm saying is this has a homotopy type of the circle. All right, so this is a B1. So you're getting a big circle here because this is empty, and you're getting a big circle. So, but this is the circle that's going to persist. Persist. It's still there, it's still there, it's still there. It's still there because I have empty space, it's still there. It's all of the circle up. Once you get there, it vanishes and it's gone, right? And maybe you have a B2, maybe you have a sphere of 3-dimensional objects or 4-dimensional objects that show up. Okay, so people put this in what called the persistent diagram, and I don't think I'll have all the time to discuss this, but instead of having a barcode, the barcode you translate it into a set of dots. It's called persistent diagram. So it starts here, it dies here. So it's going to give you this death, birth, death, birth, death, birth, death. Of course, death is after birth. So the persistent is a bunch of points that are over above the diagonal. All right. So you can replace the barcode by a persistent diagram, and then you can define a metric on it. And you can then study the persistent space as persistent diagram. Right? And so this construction, which is very clever, simple. It's basic algebraic topology. If you learn homology year number 1, but it was used in this very smart way to get many interesting results. Right. There are many interesting results. So it was used for image processing to analyze some digital images and get some interesting information. I think the most successful application was in cancer research. Data are genes, and they map these genes, somehow get some data, cloud of data, and then they apply TDA to this, and they were able in 2011 to detect a new type of cancer just using these nice ideas. Okay, right. Okay. So this is part 1 of my talk. All right, good. So part 2 is different. So you don't have to understand part 1 to understand part 2. And it's joint work with Reem Khalil and Ahmed Farhat here at AUS, a biologist and a computer scientist, and Pavel Lotko in Warsaw. And this paper appeared in PLOS Computational Biology a few months ago. So the problem here is how to classify and cluster neurons. So we want to be able to differentiate between neurons based on morphology. All right. So neurons are nerve brain cells. There are some of the data. We have 80 to 100 billion neurons in our brain. They are basically consisting of the soma, which is the cell body, and out of it go these dendrites. Right. So these are the important ones. All right. So not sure why it's okay. Sorry, there's something wrong. Okay, so here are various neuron types you can find in the brain, and biologists are interested to understand what can distinguish them in an objective manner, because you don't want to say, oh, they look different, so they are different. We want to really quantify the differences. We want to study rigorously, make it rigorous. What is the difference between these two guys? So. I don't know why this is jumping. All right, so here are names of some of the neurons you can find in the brain: the Purkinje cells, interneuron, the one in the vision. Pyramidal cells is the largest variety of 80% of neurons are type pyramidal. So they have a tuft here and they have a body. So they start like this and they go because these neurons connect between different layers of the brain. Okay, so they go from a low layer to a higher layer. So they have to have some dendritic type of pyramidal shape. Here are other types of neurons we can find. I can explain a little bit about how you make those kind of pictures later on. Okay. So the aim is to build morphological descriptors, and they have to be unbiased, stable, so not sensitive to small changes, and they have to detect some differences in families. In the literature, there are few topological or morphological descriptors. I think I only know of one. I'll mention it later on, but most descriptors are morphometric in nature. So they list the number of nodes, the number of branches, the length, the taper angles. So they take very precise measurements of how the neuron is like physically, right? And so this is put in a database called L-Measure. So you can go there and study various neurons that are put in that database, and you get a whole list of morphometric measures. And this is how we're going to deal with our neurons. So this is a picture. This is the soma. These we will call dendrites. These are the nodes. A branch is part of the dendrite that's in between two nodes. The leaf is the end. The leaf is the end of the last node, termination nodes. All right. Attends, c'est bloqué. Hein. Qu'est-ce qui se passe? OK.
C'est bon?
OK, c'est bon. Okay, so we want to get some math here. So we model the neuron N as a collection of rooted binary trees embedded in free space. So the way we get the information about the neuron, because okay, so developed the math a little bit, but also after that, once you have the math, you have to go and put it in the computer and then go in the lab and analyze families of neurons. So you really have to be able to have all the data you need about the neuron. This you get, there are databases, very interesting databases. There's one Neuromorpho that contains millions of reconstructed neurons. And these reconstructed neurons come in the form of what we call the SWC file. So the SWC file has basically 4 columns with many hundreds of points, and those columns describe a single neuron. The first column would be the 3D spatial coordinates of the neuron. Then you get the thickness of the branch at a specific 3D location. Then what is more important is the marker type. So that SWC5 tells you which one is a soma, which one is a node, all right? And then it tells you, and that's very important for us in order to reconstruct this whole thing, it tells you which is the father and which is a sibling. I mean, so if you have a node from which goes a dendrite or a branch, and then another node and goes on away from the soma, so this will be called the parent, this is called the child, all right? So we need to know immediately the child and parent so that we can reconstruct. So once you get the SWC file with all the data, you can reconstruct and get those pictures that I have given you. All right, so a sole descriptor is any rule that is a function, if you want, that associates to a given neuron. So a neuron for us then is a tree embedded in space up to O2 isometry, because of course we do not have, we don't know how to situate ourselves in space generally, but once you have a neuron, all neurons look the same if you perform any isometry to it, rotation or reflection or translation. So everywhere I talk here in this part, when I say neuron, I think about it as an O2 isometry class of an embedded tree. A neuron is a tree. Okay, a sole descriptor would be a compactly supported function into R whose independent variable, on R, sorry, whose independent variable is the distance from the soma. So it's a function that goes from from R, or a compact support of R, into some metric space. And this function has to be stable. I'll have to explain this. This is the most technical part of the work. And it has, okay, so if we normalize it, it'd be a function from 0, 1 to x. Here it is. Okay, so I wrote it here. So start with a family of neurons. And normalized descriptors then will go, it's a function, go for every neuron, sorry, for every neuron you get a function from 0, 1 into a metric space. And the interesting thing is playing with the metric space is what is going to give us very interesting descriptors. So this gives us an assignment. So I get an assignment this way, taking me from, so the family of neurons to maps from this interval into X. So to all n, I get Vn, right? This induces a pseudometric on the family of neurons, or a metric. Think of it as a metric. In fact, it's a pseudometric, but if the distance is 0, doesn't mean that the two objects are 0. That's the only difference between the pseudometric and the metric. But the fact that I am basically embedding my neurons into maps from I into X, right? So if I take a distance on maps here, I get an induced distance on neurons. Or so so for any feature, right? So for any descriptor like this, or any morphological descriptor like this, which we call a sole descriptor, I'm going to get a pseudometric. And this is the most important part of this construction. All right. So we chose to, doesn't have to be this, but this is the nicest we managed to work with, the L1 distance. So we took on the mapping space, X, remember X is a metric space. So the mapping space also has this distance, which we compute like this. Okay. So, all right. So starting with a function like this, I get a distance on neurons. Right? Now, the whole point is how to construct these in order to explain certain features. Okay. All right, let's do an example. Right, let's do an example. All right. So here's the example I want to look at. Right. So I'm going to order the branch points. So here's the soma. Here are the nodes or the branch points. Here are the radii. All right. So first, this is the first node at R1. Then this is a leaf, dies out. There's another node here. So I have 1, 2, 3, 5 leaves, 3 internal branches. So I'm going to construct this function on the neuron. So to any particular neuron, I'm going to get this is my phi. And this is the phi that's going to be associated to the branching pattern. And what I end up with is a step functions. So I start here, I don't have any branching. I don't have nodes. I start with, I claim, okay, I start with 0 at the soma. But at R1, I'm going to get my first branch point. Oops, it jumps to 1. So it continues to be 1, all right? R2, there is nothing. But at R3, I get a leaf. So I subtract the leaf. So I had a branch, I go here and subtract the leaf, I'm back to 0. Then at this one here, R4, I'm getting a leaf. But then at R5, right here, I'm getting a branch. So I'm jumped back again to 0. And then I have leaf, leaf, leaf. All right, so I have 5 leaves. So I end up with a value of -3. But again, so here's an example of what you can do. All right, so I have another, there's another possible construction. In fact, we have a few. For example, if you take the wiring descriptor. So what is that? So here's the here's your neuron. Here's your soma. All right, and what you do is you look at, for every radius, you look at the total length. So this is the simplest, one of the simplest descriptors. So around for a given radius R, I can take the radius to be a linear radius. You look at the total length of the dendrites that are in that radius, within that radius. Okay, and then you can normalize it. And so you get the total length. Okay, NR here, NR means the neuron you get within distance R from the soma. Okay, all right. So this is the.
Excuse me, Professor.
Yeah.
Assalamu alaikum. So, Professor, in the last slide, you were pointing about the distance. What kind of distance is that?
Yeah, exactly. We have two. Thank you for the asking. We have two kinds of distances. We have a linear distance. All right. So this is in 3 space, right? So I'm in 3 space, so I take linear distance. Or I can take a distance where you can do this whole thing, taking distance along the dendrite. Okay, so you take the internal distance. So you follow it's the tree distance. All right, so if you want to go from here to here, then you have to go like this, like this, like this, and like this, which is not exactly like going like this. But right here, I'm using a linear distance here. So Rn is the radius of the, it's called the radius of the neuron, right? It's the largest ball in which you can put in the neuron. Okay.
Professor, actually, to my understanding, and please correct me if I'm wrong, there are different kind of distances, first of all, and secondly, because the dendrite pattern that you're saying, it's fractional or fractal in nature. So each dendrite would be having a different distance. So how are we going to measure it, and what consideration are you taking when you are thinking of because I'm trying to understand what you're trying to explain.
It's very simple. Okay. So you think of think of your neuron as a tree in R3 in space. Okay, it's a rooted tree, so there is a soma, right? And now the distance of any point of the tree to the soma, I'm taking it's a linear distance. It's the distance in R3. As simple. I'm not taking dendritic distance. I'm not taking the distance to travel from this point, the shortest distance of travel from this point to this point along the tree. I'm just taking the linear distance. That's all. But you can do that other construction. You can take dendritic distance, right? I mentioned it here. I said, you know, if I go back here really quickly, oh gosh. Here. So I said you can get the path distance on the soma, you can take the linear distance, radial distance. So we can deal with both distances. Right? So the distance from here to here is dendritically it's going like this, and like that. But linearly it's going like this. All right. Okay. So yeah, I mean, you can do these sort of tricks, and you can construct these sort of functions. All right. And now you have a metric between those functions. So here's for example, we have compared, for example, our branching, the branching descriptor and the wiring descriptor on two different neurons. So here I'm taking a pyramidal neuron here and the stellate here, and this is the step function you get by doing the wiring construction on this guy. So you can see very easy, very nicely, you can see that because here there are many branching, lots of branching is happening near the soma. So you can see it really quickly branching off like this. And then it gets constant at some point. Okay, so you hit some leaves, so you have to subtract, but it is constant for a little while, and then you hit a tuft, all right? And then you see some branching coming up in here. This one is slightly different in shape. And so now in the feature, and this is the thing that that's been working okay for us, fairly well for us, is when you say the distance. Now, if we take the distance in the feature, in the branching feature, for this feature, the distance d phi between these two neurons is the distance, the L1 distance between these two functions. All right, and so the closer they are in distance, the more they look like in branching as in the branch pattern, the more they look alike in the branch pattern. For wiring, here's wiring. Here's very interesting. So this is a very thick neuron, right? So here's the soma, and you start doing this construction by taking, the total length of dendrites within radius r, and so you can see it, right? So it's very thick here, and then thinner, and then thicker here. So it goes like that, while this one is pretty much flat. All right, so this is what we're getting. Right, I have to go a little further now. So you can get tortuosity, another one of these features. All right, so the tortuosity is precisely what you were asking for. So if you take the tortuosity between two branch points is the linear distance divided by the dendritic distance. Okay, so this is tortuous because you see the greater the distance here along the tree between these 2 points, the more tortuous it has to be. Right. So if it goes like that, then it's fairly tortuous. So you take the ratio, which is always greater than 1, this ratio, and then take the average of ratios of something which within radius r, you get what we call the tortuosity descriptor. Right. We get the energy descriptor, which has been very useful for us because we could answer questions like polarity and things like this, which we're not going to do here. But this is I think it's a nice one because basically we put an electron here, so we put electrons here, right? Charges, right? And then we look at the electric field generated by that. So here's one here, coming from this point, and here's the electric field coming. So these are now charged electrons. And this is the effect, it's this vector field at the soma. And by the superposition principle, we add them all up, we get a vector, which is En00, if you do it at the origin. And only at the origin, this one is not, it's O2 invariant. So it doesn't matter, it doesn't depend on how you orient your, the magnitude, the vector will change if you change the neuron, but the magnitude of the vector does not. And so that intensity at the soma is what we call the energy descriptor, right? And so again, here's phi is for energy now, and this is the function he gets, right? So in fact,
Sadok, how you define the intensity here of the charge? QR?
This is the one here.
It's one, okay.
It's defined here. C'est un champ vecteur électrique. So it's depending one over the distance, right? Well, or one of the distance squared intensity. All right, here are the energy and tortuosity descriptors for a family. So if you compare this one and to this one, one, right? So here you can see the differences, and you get the distance between the two, and it's everything works well. Right? But you have to put it this way. And so I'm going to skip this one. So at the end of the day, we have in this paper, we have 9 MDs, morphological descriptors, and the branching that I mentioned, the energy. So these are the descriptors. These are the step functions we have for this particular neuron. We chose a very simple neuron because you can even trace it. You can even construct the branching yourself if you know, but it's in 3 space and it's being projected. So you have to be a little careful. The wiring distance, I discussed that. The flux is another one, leaf index is another one, tortuosity, root angle. And the other one is not a step function. The TMD is not a step function, so it's a more complicated metric space. So it's not part of this picture. And the theorem we can prove is all descriptors are well defined, obviously isometry, invariant, and stable. So what is stability? Stability is what takes a little bit of time to work out, but it's very simple. You want that if 2 neurons seen as trees, all right, trees in space. So if they are close in distance, so they are, so a neuron, we actually view a neuron as a, we see, how can I explain this? Right, so we have to view a neuron as parametrized. So all the branches are being parametrized. So smooth maps, smooth paths in space. And so we can take a distance between them, and also we can take bending and curvature of the neuron. And there's a metric that's used in knot theory, which is called the delta theta metric, that we thought was the most interesting. And that two curves or 2 neurons or 2 knots are close if pointwise, you know, they're close at parameter t, and the tangent vectors are close, right? So they don't bend too much. And so using this delta theta metric, we call a Scholl descriptor to be stable if 2 neurons being delta theta close, it's the continuity, I mean, the usual continuity. Then their distance for that Scholl descriptors is less than epsilon. So if you have a control on epsilon, you can get them close enough so that the distance in that branching feature is less than epsilon. All right, so
Professor.
Yes.
So Professor, say for example, if I'm having a wireless connection, I'm having an antenna with me, and I'm trying to
you have what? Sorry?
Can you hear me now?
Yeah. Yes.
Okay. Okay. So Professor, say if I'm having an antenna with me and I'm trying to interact with someone over antenna, let's say I'm trying to get in touch with you, and so
please, can we suggest to finish the talk, and you can ask your question just after the talk, please.
Oh, okay, sure, sir.
Okay, thank you. Thank you.
All right. So stability is a technical part of this, but that makes, I think, what makes the theorem interesting. This is something that we noticed recently, and it's not published, but it's very interesting if you use the Skorohod metric for the step functions. So you notice that the step functions are type of a Cadillac functions for those who do probability theory. So this means continue à droite, limite à gauche functions. And so for most, in fact, 8 out of the 9 descriptors we have, this association takes a neuron and associates to it a map, which is a step function. Right. So it's a step. Because you have step functions, you have, sorry, I cannot write with this. You have another metric, it's called the Skorohod. And the beautiful fact here is that it made our lives simpler. We did not know about the metric before, but it makes stability simpler to prove. All right. So the Skorohod metric is really magnificent metric for people who don't know it. I mean, I see here is some functions that are very close, right? If you take the soup, right, here's the blue function and the red function, their distance, if you take the soup of f of x, g of x, then obviously the distance is 8, right? So there's a point here and the point here whose value, the difference of values is 8. But if you allow yourself reparametrization, this is reparametrization. If you allow yourself reparametrization before you take the soup, that's how the score-Hod metric works, then you get a distance of 0, 1, which is what you want. Right. So it's even a better metric. All right. So really quickly, I don't know, do I have 5 minutes, Nabil?
Yes, yes, of course, yes.
So some of the applications, the applications are for classification and clustering. All that I have been discussing is not all that interesting to biologists. This part is what biologists are after, and this is what perhaps makes the paper interesting. So you can do just standard clustering on data. So I have 2 data sets. We had actually 4 on the paper, but this maybe the simplest ones are there. So we took a total of 30 cells. So these are the Purkinjes here. These are the interneurons, and these are some medial prefrontal cells. Right. So we took 30 of those, and then we run the morphological descriptors on this. So what do I mean by that? All right, we did what we call hierarchical clustering. So you just basically take a feature phi, and you take the distance that is associated to phi, and or phi, sorry, in English, and you take the distance matrix. All right. And then using the distance matrix, you can, of course, do the usual dendrogram. So it's very nice that for this particular family, we took 30, 10, 10, 10 of each. So, okay, let me go back. I'm sorry, maybe I did not explain. So what I want to, so I took 10 of these. So I took 10 of these. I took 10 of these, but of course I can take many more, but just 10 of these. And now I want to run my descriptors, and I want the descriptors to tell me what is the difference between neurons of type A, neurons of type B, and neurons of type C. All right. So are they more tortuous? Yes, maybe the left ones are more tortuous. So let me run the tortuosity descriptor. The wiring is certainly going to detect this is thick, this is thick, this is not thick. But there's the one that I haven't discussed that they call TMD was actually extremely good for this family. So we did run this and we took the distance. So you take the 30 by 30 distance, because you take the distance between any 2 points in the family of 30. And you take the distance matrix and you do k-means, or you do whatever. So you get the dendrogram. And so they are really split into three families. The ganglions are here, the Purkinjes are here, and the interneurons are here. Right. So this descriptor here, the TMD descriptor, was very good. It's metric. I mean, of course, we use the metric associated to this to give us a separation. So we managed to separate them based on this TMD. We also can do something like this. All right. So I need to see, maybe jump here. Okay. All right. Let me explain this in through the pictures. We did this. So we took this family here. Now we were able to separate them, but now more interestingly, we wanted to compare our methods to all their methods. So L-measure method means what? You take any neuron and associate to it a vector. The vector is made out of numbers, morphometric, all right? So this is a completely unsupervised kind of so you vectorize this neuron by associating to it a bunch of numbers. The thickness of the stoma, the distance between branches, the volume of this, the volume of that, the distance of this. So all things that people measure, you put them in the vector, right? We had the vector with 24 entries. And then you take, you have vector in R24, you take the Euclidean distance. So you get a 30 by 30 distance matrix with the Euclidean distance. And you look at the dendrogram that is associated to this distance matrix, and you see that the usual, if you do the L measure technique, it's not very, it doesn't really separate the family, right? These three things. Using our shell descriptors, we were able to completely segregate them in a very nice way. All right. These are topological. These are morphometric. This is morphology. So our vector here, we also associated a vector to this. All right. But the vector is quite different, right? All right. So this is about the area underneath our functions, some derivatives, things like this. So morphology is working better than morphometry here. All right, another example with tortuosity. Just really quickly, the Martinotti cells are known to be tortuous. People know that kind of tortuous, right? So we run our descriptor here, and we very much managed to segregate them, right? All right. So for the tortuosity descriptor, this is the dendrogram associated to that, and you can see that the Martinotti cells are close, means that in tortuosity they look the same, and they are far from the others, which means that they are probably more tortuous than the others or less, but the distance between the features is clear. All right, finally, the classification. All right, so, okay, so this is the last part. We classify the neurons. So what's the point here? So if you give me a certain number of classes of neurons, which you know, so it's a supervised setting now. We're working with a supervised setting, and then somebody hands me a neuron that I don't know, right, and tells me in which class does this neuron belongs. Is it from the vision cortex? Is it from the some part of the brain? Is it vision? Is it interneuron? What could it be? So we managed to answer this question as well to some extent. All right, so so what do we do? So you start with our family of shell descriptors, right? So these are the shell descriptors. So this is branching, this is wiring, this is tortuosity, etc. This is TMD, whatever, right? So you have a number of shell descriptors. And now you have K classes, all right? So you have the interneurons, you have the ganglions, you have the Purkinjes, you have the vision, something like that. So, all right. We are going to associate to this data, to any neuron in any one of these classes, or any neuron anywhere, a vector. So we're going to have a supervised vector here. So we have the data, we have the omega, we have the C's. Out of this, we are going to associate a vector to any neuron, right? So the vector is going to be given like this. All right, it's this vector which is based upon, you don't want to spend time maybe trying to understand how you vectorize this, but it's very simple. You take the average distance, so you take any neuron n, and you take in, okay, because we know we have a metric for any shell descriptor, we have a metric, right? So we can take the distance from n to any n prime in this class and take the average of it, right? So this ith entry is going to be this average distance. So you put the average distances, right? You have K, for every feature you have K classes, so you can have K average distances for phi1, and then you go for phi2, and then for phi m, you have still K average distances on this distance. All right, so you have now a big vector in R to the power mk. All right, so where is this? This is in R to the power mk. All right, so what have we done? We have taken, for a family of short descriptors we have, for a certain number of classes people look at in biology, they have these classes, right? And now we're going to come with our technique. We're going to tell them, all right, give me that neuron. Sorry? Hello?
Yes.
Oh. Okay, now I thought someone asked. I'm almost done here. So, given a neuron n, I can construct a vector in R to the power mk. All right, now what am I going to do with this vector? All right, so here's a situation. We took a data set with 300 neurons, right? Now you have these vectors in a big space. You do PCA, principal component analysis on this. You project it into space, and in general, most of the time, because you're dealing with a big number of neurons, you're not going to get good separation. So the vector we constructed is not optimal right away to give you separation. And this is now the neat idea. That's the end of this work here is you don't have to do Euclidean distance. You don't have to measure the distance between vectors as your Euclidean distance. You don't have to do this. In fact, people have invented a beautiful technique called metric learning, which constructs a metric. It comes up with a metric that is suited for your classes. All right. So for example, this is a nice technique. So if you have, say, blue and red points in space, in Euclidean space, all right, and they are mingled, right? So the distance between the red and the red could be big or small. The red and the blues could be big and small. Well, what you want another metric such that the distance between the red and the red is a bit small, because you want them to be close. You want the distance between the blue and the blue to be close, and you want somehow the distance between the red and the blue to be far, right? So you want to come up with a metric, if you can, based on the distribution of these points that separates the classes. This can be done to a good extent by this crude idea, which is to construct this metric here as so the distance between two vectors is not VI transpose VJ, which you know, the Pythagorean distance that you teach, but you're going to insert here a matrix, positive matrix M, all right? And so when this M here is going to change the metric. And this is the M you construct from the data you have, so that you make the blue and the blue close, the red and the red close, and the blue and the red far. All right. So one way to think about this is there's literature. You can look at it. There are papers written about how to construct M. It's very heavy linear algebra. But basically what people, the idea is that your M is of the form L times L transpose. And so it's basically you take your vectors in Rn, you transform them by a matrix L, and then you take the distance then. So so the new distance, it's called the Mahalanobis distance, is now the distance between the transformed VIs by a, and all right, so this is what we did. This is our main reference, metric learning in Python. So we had to take this instruction from them. And so we did run successfully the metric learning. So we constructed the new metric, right? So yeah, so sorry, I need to really quickly tell you how we did it. All right, here. So you see, this is the data here. This is the data, 340, I think, some neurons we got. In 5, 1, 2, 3, 4, 5, we have 5 classes, right? So here K equal 5, right? And we had, I think, features-wise, I think we had maybe 6 or 7 features. So we are in R35, and then you project them out using this PCA thing, and to get something that look like this. Now, what we have done is PCA uses Euclidean distance. We changed this, into a using this Mahalanobis instruction, we got a new metric between the classes. We use now this metric to vectorize the neurons again, as we did before, and then we projected them out, and we got a good separation. And in most data sets, if not all data sets, we use this technique. It was definitely better than just going straight. So you really have to, you do the metric learning, and now you can do your classification. We're done. So you bring in an arbitrary neuron. So we brought one that is granule type. So something more belongs to this. You take its distance to the classes, and okay, so here we got it here. We saw that it's far from here, far. So it has to be of the granule type. So we tested it. Somebody gave us a neuron. We told them right away this guy is a granule, and because we tested it on this scheme.
Okay, thank you.
That's it. We're done. I'm done. So this is a pipeline. This is the whole paper. This is the construction. These are all descriptors. This is how you do the whole thing. You get the SWC file in order to understand the neuron, right? You generate the shell function. You compute the distance. You generate distance matrix. Then you do the dendrogram. That's for the clustering. If you want to do the classification, you do the vectorization, then you do the metric learning, and then you do the classification. So I explained everything in one diagram. Further work maybe to deal with tree-shaped structures in lungs, in eye, in anywhere you want.
Thank you, Sadok. Thank you for this beautiful talk, because you give us the opportunity to understand how we can use topology on these topics, and it was really interesting. Okay,
thank you, Nabil.
Is there, I think, Patrick, if you want to ask your question now.
Now I forgot the thought, actually. I was trying to ask something related to lightning that we generally see, and the pattern that it actually recognized with one of the patterns that professor was sharing on the slides. But right now I forgot that question. Sorry.
Do you have other questions, please?
I would have to read his paper first. So certainly I'll look that up.
I can invite you to see papers to these two journals. I always conclude my talks by asking submissions, requesting submissions to these two papers.
Okay. Thank you for sharing this. No other questions?
Professor, could you repeat that? What were you saying last, that you end your talk with what? Say
it again.
You were mentioning that to submit some submission.
No, I'm soliciting submissions for these two journals. I'm an editor in both. This is the Tunisian Journal of Math. It's very advanced. So this is an A-star journal, and this is the Graduate Journal of Math, which is also Scopus, and it's a really good journal as well. Right. So if people are interested to submit papers, are most welcome.
Okay, thank you.
All right, I mean, it seems no questions.
I told you that the attendance is special, especially from stochastic processes and partial differential equations. Then I think that your topic is of course interesting, but something close, not close for our.
Yeah, no, I understand. Of course, it's always.
But for me, it was really a good thing to follow this talk.
But you see, Nabil, if you want to do any kind of data analysis, right?
Yes.
And I think you're interested, and I think you even gave even school in Tunis at some point. So how do you deal with data, right? In the case people who are interested in working with data. So I mean, you take data.
I'm not sure. I'm not sure I understand your question. If you ask me,
yeah, I mean, what
how
are
I
the
deal
techniques?
with
Yeah, what are the techniques? You know, what is the mathematics you use in order to understand data, and what does it mean to understand data? What is data analysis?
Okay.
Right. So
you know, there is many techniques of machine learning. Okay, then there is a lot of techniques, and now there is a lot of papers that use deep learning, and we have a discussion together, if you remember.
yes.
And I explained how it works, and the deep learning is a technique that can be used for any kind of data, okay, and even for learning.
How do you get to represent?
So you can learn your neural network, your deep neural network, for some data, and you can use your neural network for another kind of data, and it works. But the problem is that it's how you explain that it works. And
yeah, no, I mean, Nabil, sorry. I mean, for example, I'm now asking the questions. For example, I go back. How do you deal with data that comes as neurons? Can you do you vectorize them? Do you transform them before you can deal with them?
I suppose that the general way to deal with these problems are with pictures. Okay, then it's just image processing. Okay, then they deal with these problems as just a picture. Okay, and they have thousands and thousands of pictures, and they process just then they have convolutional networks that are very efficient for images, and I think that they can make the data analysis with the pictures of data. But I am sure that your technique, or these technique that you present, okay, are more explainable, okay, and because you have features, you define features, okay, and what is difficult sometimes is how to define these features, okay.
Yes. Yeah.
And
yeah, because you know, it's you know, we have to, in my opinion, you have to transform the data into something you can work with.
Exact.
And the data, the topology, it's not something like I can hope to use maybe differential equations, because people love differential equations or something. Because I would be tempted to define a space of neurons on which I can do some flow and some topology and some differential equations. There is no such a thing. I've been trying to think about the way to introduce some kind of a differentiable. First of all, I have an interesting space of neurons on which I can do some differential equations, for example.
Yes, I don't know if this is possible or not. Okay, but there is a lot of examples where we don't imagine that we can use partial differential equations, and some people did. And
no, I mean, okay, I'll look saying differential equations, but I'm related so that you relate the space. So to me, this was interesting, very interesting, because you go from abstract data. You understand, Nabil? I go from abstract data to something that I can manipulate.
Yes.
Right. And because it's topology, you have to go through space or functional space or distance or a metric space.
Okay,
All right.
is there other questions or remarks?
Yes, Dr. Nabil.
Yes,
I have a question. Okay, thank you, Dr. Sadok, for this great talk. I have a question concerning classification and clustering. Do you have used the machine learning algorithms?
Yeah, I think we did. Yes, the technician there is Ahmed Farhat, and I think yes, we did.
Okay, that you take the data set and you apply all the and you have cleaning the data set and scaling the data. Then you apply the clustering and classification machine learning algorithms methods.
Yes,
or...
I was going to show you one main website that we used for the classification, and it's all machine learning. I mean, it's all machine learning.
Yeah,
there's no doubt about that. Where is it? This one. Maybe you want to copy that.
okay. Yeah.
This one for classification. The code
and clustering also.
No, well, clustering is k-means, like very simple.
K-means. Okay.
Yeah, we used dendrograms for just general. You take a metric distance matrix, and then you compute this.
Yeah, I know that. Yeah. Okay, thank you.
We did not use anything complicated for clustering.
Okay.
We defined something like detection. It's in our paper, as a notion of percentage of how much each feature is going to detect. So if you have a family of neurons, we wrote a code for this. It's not in the literature. So you want to find the family of the most tortuous, for example, bunch of neurons. So we detect the tortuosity in terms of percentage.
Okay, thank you. Thank you, Doctor.
You're welcome.
I have another question. I think I missed something in at the end of the talk. You explained that you made a learning metric. Yes, this is the slide. And then you mean that your metric was built from the data. Is it
yes
correct?
Yes, from the classes, from the
okay.
So it's supervised. We mentioned it's supervised.
And which algorithm you use to make the metric learning?
Yeah, this is what I shown to Mrs. Khawla. So
okay.
This is exactly.
Don't you say it? Kevin?
Yeah. No, it's not. That's for clustering, but for classification. So the metric. So I have K classes. So I start with the number of. So here, maybe here. My gosh. So I start with the number of pyramidal cells, a number of granule cells, ganglions, right? So I have five classes. I have over 300 neurons. So maybe 40, 50 from each, or 50, 60 from each. And then I use I vectorize them, right? And then I use now, once they are there, I use metric learning in order to get a better metric, a D metric, that separates the classes.
Okay.
All right. So the D metric comes after you vectorize, and we use some big database here, repository of metric learning Python. They've done an amazing work. I mean, there's people who are working. Many are in Riyadh, in fact. It's very, very nice.
Okay.
Right.
Okay.
Yeah. So this is for classification.
Okay. Okay. If there is no more questions, perhaps you can
just a general question. Thank you, Sadok, for this nice talk. I have a general question, maybe outside the topic of this talk. Can us classify the families of neurons as known families of trees, like a binary tree or recursive tree or else?
Well, yeah, I mean, neurons are a bunch of binary trees, which have a common root. All right. So here's it's called the primary. This is called the primary tree. All right. So here's a node. Here's another node. Here's another node.
Yes.
Okay. So this is these are internal nodes.
But okay.
And
yes.
These are the data leaves. Now and then you have another primaries. You might have another primary like this, and then you may have another primary like this. But the interesting thing that it's a binary tree. All of them are binary. So the branching, it only branches into two. You cannot have something like this.
Okay. No three branches. Yes.
That does not exist.
Mm-hmm.
Yeah.
Just on the root.
Yeah, and so it all starts from the soma.
Mm-hmm. Okay.
In fact, in the L measure that I discussed, the number of primary trees exist. These are morphometric data. So they tell you how many of these primaries exist. Right.
Okay.
Yeah.
There is another question. Gobina, France Gobina.
Yes, thank you, Dr. Sadok.
Welcome.
My first question is on the different shows descriptors. Can we define an equivalence relation on all of these shows descriptors and come up with an equivalence relations? And what would be the significance of this equivalence relation on the shows descriptors? And my second question is, can we replace the H2 holes with open balls or closed balls? Thank you.
Yeah, no, of course, I can answer the second one easily. You can use closed or open balls, not a problem. It works the same way. I'm not sure if that was the question.
Okay.
Now, let's see. About equivalence, I don't know. What do you mean by equivalence class? So descriptors are functions on the space of neurons up to isometry. Not sure. Why? Why would you want to do that? I don't see anything that would be of interest to do such a thing. I mean, I'm not sure what kind of yeah. Hello? Hello?
Yes.
Okay. All right. Yeah, no, sorry, Mr. Gobina. I don't know. I don't have an answer to your equivalence class question. But yeah, no, you can replace open balls by closed balls, and you can even do some convex things if you want. But closed balls and open balls are okay. Okay.
Okay. Is there other questions?
Oh, I see that Walid bin Hamouda is there. Hi, Walid, if he's listening to me. All right. Yes. So thank you, Nabil. Thank you very much.
Okay, thank you for accepting.
Thank you, Rafiq. Thank you both for this beautiful opportunity. And I hope somebody got something out of it.
You are welcome. You are welcome. It's
okay. Okay, good.
Merci,
Fantastic.
merci Sadok, et à très bientôt. On se rappellera de toute façon.
Bien sûr, avec grand plaisir. Attends.
Ok.
À bientôt.
Oui, c'est mal.
J'essaie d'arrêter le stop share. OK.
La Sadok, ça va?
وسام شنو حوالك؟
Et malheureusement, ma cam n'est pas fonctionnelle, donc tu peux pas me voir.
Thank you, Fatih.